morning, everyone. So, as we studied the Apomixis in the last lecture, and today in this lecture, this is your 32nd lecture, and today we have to study the classification of Apomixis. So, basically, whatever we have been studied about Apomixis, now we have to see what are their sub branches and how we can classify them. So, basically, here you can see. Let it draw. Let me just adjust it. Okay. So if we see like what is apomixis, so basically as a plant reproduced by several means. Like there can be a sexual method, asexual reproduction, vegetative reproduction. So basically apomixis consider under the asexual reproduction means, and this is very different from the amphimixis process, which is basically the sexual reproduction as we already discussed in our previous lecture. So this is just for your background check that you didn't miss what we have already studied in our last lecture. So now there is some point, or you can see the learning objective of this following lecture. Will we have to study about the meaning of parthenogenesis, Epigamy, apospory, Adventive Embryony. So these are the following types of apomixis which we are going to discuss in this lecture, and also their type, like the subcategory. Like the first category, we have to classify the apomixis. There are some basic factors which we will discuss in this chapter, like on what basis we are classifying them here. So this will be followed by later. So to understand importance and role in plant breeding means how the apomixis process is very helpful in conveying the plant breeding method or how they are maintaining the genetic dirt. So these are the basic productivity thing, or you can say basic objects we have to take under consideration while we are starting this lecture. So next we see here, like if we're talking about mode of reproduction, that plant undergo several mode of reproduction. Basically, though two mode of reproduction are basically fundamental. First is sexual reproduction, and second one is asexual reproduction. As we already know that sexual reproduction require the gamete production and also the fertilization process, but apomixis, which considered under the asexual reproduction, is totally opposite to the sexual reproduction. So it is totally different. In this type of mode of reproduction in plant do not undergo the meiotic division. Okay, so this you have to be studied very carefully. Okay, some people get confused that they also write the meiotic division in the apomixis. So please take it very carefully that meiosis will never go under the apomixis process. There will be only one kind of division that will become under the apomixis, that will be mitotic division. And I already told you about meiosis and mitotic. Like meiosis is a reduction division, okay, and that happens only in the case of sexual reproduction. But in the case of asexual reproduction, the vegetative method or apomixis, yeah, other methods which come under the asexual method, they never go under the meiosis process, because at this time cell is only in the place of 2 in number. So you can say the cell will be in a diploid format. 2n chromosomes mean full number of chromosomes will be present at the time of asexual reproduction. That's why these cells do not face any kind of problem and they easily propagate and form a new plant. So you can see here asexual reproduction take the apomixis under that, and but also the apomixis have gametophytic and sporophytic stage, but their gametophytic stage also not combine the sexual reproduction. By the sexual reproduction have essential key of gametophytic generations, and also if a plant which are having the apomixis that is not very rigid to the apomixis, they also have the gametophytic phase in the plant, but they do not involve the fusion. So you can say the strictly prohibition here is about the gamete fusion. If the gamete fusion take place, then there will be no apomixis. That will purely the sexual reproduction. So this is hence very carefully you have to understand it that apomixis never gonna accept the fertilization process in their mode of reproduction. So next, you can also basically compare the sexual reproduction and apomixis in this picture. Like you are seeing here, there are the two diagrammatic representation of mode of reproduction in plant. Okay, so first one you are seeing the outer circle. So if you see in the outer circle, this is the pattern of the sexual reproduction. Okay, this pattern that the ovule, that is the female part of the plant, and that goes under the meiosis division, that is basically the reduction division, and they will be formed haploid megaspore, that is gametogenesis process started from here. So haploid megaspore, then there's the megaspore gametophyte, and after that, pollen will come here. They get fused, double fertilization happen. Hence, the product will be formed in the case of zygote, and zygote will be converted into the embryo. So this is the very basic, or you can say this is the fundamental process by which plants get reproduced. This is called the sexual reproduction. But here, as we are talking about the apomixis, so apomixis is quite different from the sexual reproduction. How? You can see here, in this is a ovule, the female gametophyte, female part of the plant. So hence that there the division will be taken as a meiosis way, but in this case, in the apomixis case, there will be a mitosis division. Okay, if the division is mitosis, this is the first stage, and this will define the process will gonna be a sexual reproduction or it will be on a apomixis phase. So here you can see it that mitosis will create a diploid cell. So basically, that you already see it written here, unreduced megagametocyte. Okay, this is a gametophyte but unreduced, means the chromosomes number are not reduced here because that is the feature of meiosis. But here the division has been taken by the mitosis, so that's why the cell is diploid, and you can see here that mitosis without fertilization, that mitosis division will be happened during the embryo formation, and also when it got developed that only divide by the mitosis process, and mitosis will lead into the formation of embryo, and embryo will be converted into seed, then seed will be converted into a new plant. So this is the general phenomena between the process apomixis. This already we discussed it in our previous lecture. Now our focus is here on the classification of apomixis. So, as we are talking about the classification here, so there are three basics, or you can see, you can call them. They are the baseline on which we can classify the plant apomixis process. So, first process here, or you can say the first involvement or the first base here is cell involved in the formation of apomixis. Okay, so the process apomixis, which kind of cell is involved in that particular process, on that basis we classify them. Second is occurrence. Like if you're talking about the occurrence, then occurrence is like if the plant following the process of apomixis in their next generation or not. On that basis, we can classify them. Third basis is frequency. Now, like how frequent the apomixis process is happens in a particular plant species. So these are the basic. You can say these are the base on which we can classify our apomixis. Now, next we will see what kind of process are here. You see, if we are talking about the basis of classification on the basis of cell involved, like what kind of cell involved, then the type of apomixis is parthenogenesis, epigamy, apospory, and adventative embryony. So these are the four basic type of apomixis on the basis of involvement of the cell. So if we see their brief description here, So parthenogenesis is that the embryo developed from the egg cell. Okay, if a embryo will be developed from a single egg cell, then the process will be known as parthenogenesis. In a case of apogamy, embryo develop from either synergis or antipodal cell. So as the embryo sac having these all cell inside them, like the synergis, antipodal cells, oligonuclei, egg cells. So basically, from which of the eight cells, or within from which these eight cells, an embryo going to be produced in a diploid manner. On the basis of that, we are classifying them here. So apogamy, embryo develop from either the synergids or antipodal cell. Okay, synergids can be basically their role, or the antipodal cell can play a role for the formation of the embryo sac. Apospore, if we are talking about the apospore here. So in this case, embryo develop from the embryo sac, which has developed from the cell of Archisporium, mucilus, or integument. This is a little divergent apospore, because their embryo will be developed from the embryo sac. But here the problem with the embryo sac only. So embryo sac is basically developed from either the cell of the Archisporium. We will discuss it in detail later, so don't get confused here. So it can be developed from the archisporium, or it can be developed from the nucleus or integument. So these are the basic, you can say these are the basic type which take under consideration like how or what cell will be involved in the formation. Second base here is the occurrence. So recurrent apomixis and non-recurrent apomixis. These are the two basic kind of apomixis which basically present on the basis of occurrence. Like recurrent apomixis, if the embryo sac has diploid cell and embryo developed from the diploid cell. Okay, so this embryo sac development from a diploid cell, that is called the recurrent apomixis. It means like if a plant having a apomixis process in their lifespan, and they are generating the seeds which have been formed from those diploid cells, and having a generation of the plant having same exact character and following the apomixis process forward. So this kind of, like you can see here, the process in the continuation. Okay, so like the parent have followed that process, their daughters will be following that process. So if we are seeing this kind of pattern, well, okay, if the pattern is going to be followed, then this can be known as a recurrent hypothesis. But if the pattern will be break, or you can say that the cell was haploid. If the cell was haploid, the 70 to 90% chances are there getting for the sexual reproduction. So if the cell are diploid, there will be 100% chances for getting the apomixis process. But if the cell get the haploid division, so there will be a more chances of getting the sexual reproduction. So that's why non-recurrent apomixis, in this pattern, we are not very sure about that their daughters are going to follow their same pattern as being their parents have been followed. So third one is the frequency. So this there you can see two type of frequency. On the basis of frequency, we can derive them in the two manners: obligate apomixis and facultative one. So if we are talking about the obligate apomixis, so in this type, if a plant having only the apomixis process for their reproduction, okay, there is a plant and they are only having the apomixis for their reproduction process, this will be known as a obligate. But if a plant which having the apomixis, but in the addition of apomixis, they are also having the sexual reproduction. Okay, we are getting the point. Like they are having apomixis, but their apomixis may be induced by sexual reproduction did not take place very well, or maybe they take simultaneously place inside a plant body. Like there is a sexual reproduction, and there is a apomixis. So at that time, we call them the faculative apomixis. So I think you don't have any problem with this point. Like you got the parthenogenesis, apogamy, apospore, or adventative embryony how to take place. So we have to, on the basis of origin. So in today's lecture, basically we are going to cover it like. parthenogenesis process, apogamy, apospore, and adventative embryony. So let's take a look on parthenogenesis. This is very important, and in the most of the plant, basically this is following. Even if they are not naturally present, we are inducing them artificially, because you will see when I will going to explain the advantages of these. So you will get to know like how much they are important in today's fruit or organic food. So this is very important process because they are the basically carry forwarding the pure lines, because as a diploid cell get into the seed conversion in a plant just formed by a single parent. So the all the character which are having their parents and transferring to the offspring, they will be same. There is no mix up. So if any fruit like there is, if we're taking an example of a tomato plant, so if there is a tomato plant and they're having a very good taste, they are having a very good size, they're having a very good color, so why we want any interpretation or any mixing in that variety? Okay, we don't want to be any disturbance in that particular gene code. So at that time, we need these kind of process like parthenogenesis, apomixis, sorry, apogamy, apospore, because their main motive is to conserve the purity, conserve the genetic purity. So that's why we also introducing these things in a artificial way. This can be happen in a natural way, but also as we are doing it artificially to get more profit in our economic buildings. So take a look on the parthenogenesis. So as a term, parthenogenesis was coined by Owen in 1849. It's an ancient one, okay? Not much ancient, but a little ancient and then the modern process. So we cannot call it a modern process. We know it from like 2 centuries. So it refers to the development of embryo from the egg cell without fertilization. So this is the principle of apomixis, that they do not get fertilized. So basically, in refer to development of embryo, so embryo will be developed without the process of fertilization. This is common thing. Parthenogenesis has been reported in several crop plants, such as there are some example in which parthenogenesis have been seen, or you can say we can artificially introduce in these kind of crop plants. And basically, the crop plants, if you compare the crop plants and the fruit plant, okay, if we are taking an example like there is a crop field and there is a crop fruit, so parthenogenesis is most common in the crop fruit as comparison to the crop fields. I will tell you why in the next when we study the induction process of parthenogenesis. Also because if we compare, okay, we will discuss it later. So parthenogenesis is of two type. If we see the types of parthenogenesis, there will be two type of parthenogenesis. So basically, this can be the haploid parthenogenesis, and second one is can be the diploid parthenogenesis. So diploid parthenogenesis and haploid parthenogenesis. This is based on it what kind of cell they have been developed. Okay, there are some important point of parthenogenesis. You can read it out. Like I can read it out for you. The embryo develops from the egg cell. It's obvious. That is the main core of parthenogenesis. That they have to be developed from the egg cell, and this should be deployed or can be deployed. So most of the time they are deployed. So it may be either haploid or diploid, but most of the time this is diploid. It is genetically controlled. That is a very certain situation here that they have to be genetically controlled, right? So that's why they are having this process of apomixis. Otherwise, if there are no genetic control, so there will be no apomixis will take place. It occurs in nature as well as can be induced artificially. It is useful for conserving heterosis, obviously. Like, what is heterosis? Like, what you meant by heterosis? So, heterosis is basically the best hybrid character. Okay, if we generate a plant with the best characters and they are showing that particular character dominantly, so we wanted to secure that, okay? Everyone if we are implanting a particular gene for a specific character, like for take an example, like we want a fruit to be look pink, or blue, any color. So if we wanted that to be look blue or pink, or we specify a particular gene and induce that gene inside their body, and if they are showing their feature in a phenotypic version, okay, there will be two side, genotype and phenotype, okay, in a genetics. So in genotype, we just induce a gene that is the internal working, but if we see their phenotypic effect, means if a gene is enough capable to express in morphology, so that is a hybrid heterosis expression. So basically, in the process like parthenogenesis, we can genuinely conserve them. We can conserve these kind of processes. So that is very good. Like we are having what we want, and we can carry them forward by these kind of processes. Next, it is useful in developing floor lines. That is very obvious because they do not conserve any kind of mixture. Okay, because there are no fusion of gamete take place. There are no several other character will get introduced. So that's why they are developing the pure lines. The haploid parthenogenesis that is very efficient in developing the pure lines. Why? Like if you're talking about heterosis, like we talk about heterosis, that is the major product of diploid parthenogenesis. Okay, so diploid parthen in that type of diploid parthenogenesis, not all the character has been shown in the phenotype. Okay, some character have to be suppressed and some characters have to be show their effect. That is the heterosis basic phenomena. So, but if we are talking about a haploid parthenogenesis, they assure us to give us the pure line because haploid is all about the half number of chromosomes, and all these half number of chromosomes have to be perform their work. So all the things which are present inside have to be present outside. So these is a very as like promising one. That what is inside, what is outside. Okay, like again, if you are enough good from inside, show it outside. So this is that kind of thing. Like they are really pure lines. But deployed one, they conserve a little characters and show what is dominant. So haploid parthenogenesis basically develop from the haploid cells, not very much to elaborate. They are just responsible for the pure line because there are no chromosome doubling happens inside a haploid parthenogenesis. So that's why whatever they have, they are displaying them. Next is diploid parthenogenesis. So, as we know that diploid path, as the name suggesting, they develop from the diploid egg cell, and if they are developing from the diploid egg cell, they will be called as diploid parthenogenesis. As the definition suggests here, sometime embryos develop from the reduction division, like egg cell, antibodal, synergid, and such embryos are diploid. It gives rise to diploid embryo. So, sometime what happened here, like maybe the reduction division will take place in a very in a preliminary time when the embryo has been going to in a developing phase. Then at that time, maybe they are developed from the reduction division. But as the time carry forward, maybe there's something happen when the haploid cell become diploid. But this type of diploid embryo and this type of formation of diploid embryo or the process diploid pathogenesis, that means you can say this is rare, this is not a common process, and they are reported in grasses like terexum and basically, but is very useful in conserving the heterosis. So these are the basic some diploid pathogenesis. There is also one another term you can see here, androgenesis. So androgenesis basically at that time pollen came to the embryo cell. What happened at the sexual reproduction? Pollen have to become to the female flower to the stigma. Okay, but at that time pollen come, but they do not induce a sexual reproduction. But in the place of inducing sexual reproduction, they induce parthenogenesis. Okay, there's some type of chemotaxis process that have been done by that pollen, and when that pollen come here doing some chemotaxis process and inducing the parthenogenesis in that particular plant, So that type of parthenogenesis is known as the androgenesis kind of parthenogenesis. This is also rare, but we can artificially introduce them in tobacco, rice, and some other crop species. We can do that, but naturally, it is a very rare phenomenon. So now we talk about what are the causes of parthenogenesis. Okay, so if we're talking about cause, like what things is basically introduce the parthenogenesis in the body. So there are several things like inability of pollen tube to discharge the content inside the pollen embryo sac. Now, if you're whatever the inabilities you're going to see here, the most of the inabilities are depend on the inactivity of pollen. Pollen will come, stigma. Maybe there are several factors which do not get led pollen to get inside the female part, and other factor, other morphological adaptation can also help, cannot help the pollen to get inside. So, because of inability of pollen inside a female part, reversely it induce a parthenogenesis. Okay, so these are the proportion thing. Like if the pollen is not going to perform, then the most of the chances is getting about the parthenogenesis process. So we will take it one by one here. Like pollen tube, they discharge the content inside the embryo sac, means they cannot enable to discharge the male gamete inside. Insufficient attraction between the male and female gamete. So whenever the male and female gamete come close, they have a chemotexis activity. If that chemotexis activity is not sufficient to get them into each other, so then also parthenogenesis will take place. Early degeneration of the sperm. Maybe there are several factors which can possess the early degeneration of the sperm. In that case, also parthenogenesis happen. Very long style, as the style is a part of female reproductive organ. So this is the path which pollen have to be crossed when get to the female bud or egg cell. So if a style is very long, so sperm have to take a little long time to get to the egg cell, but in that case that can degenerate in a plant. So at that time also parthenogenesis happen. Next is the sclerenchymatous style. So I already told you about the simple tissue, parenchyma, collenchyma, and sclerenchyma. So basically, sclerenchyma are the tissue which have a very broad lignin on their walls. So if anything which have a very heavy deposition on their walls, so basically what they do, they block the way. So these deposition fill up the gaps in the stylostigma. So that fill up road cannot make or you can say when the pollen comes, so they cannot find a way to the embryo sac. So at that time also parthenogenesis happen. Short pollen tube formation. When the pollen tube form for the pollen transport, if that is very short and cannot reach up to embryo sac, at that time also parthenogenesis is induced. Slow rate of pollen tube growth. If the growth of the pollen tube is very slow, so obviously pollen get basically they get degenerate because pollen have a life. If they are already activated, they have to be reached to the embryo sac in a time. But if they are not getting there in a time, they can be deactivated. So these are the very important, you can say these are the very fundamental causes of performing the parthenogenesis, like stimulation of pollination in absence of pollen tube. This is also a very important, you can say this is a very charming one, pollination happens and pollen come to the uh, place of the female flower. But at that time, after even getting at their destination, pollen tube, if not present there or pollen tube is not formed there, then again the pollination happen and that pollen which come to the fertilization process get away in another plant. At that time of situation also, pathogenesis happens. Self-incompatibility or cell cross-incompatibility. Plant reproduce pollinate by two methods, self-pollination or cross-pollination. If they are incapable of transferring the pollen, in that case also pathogenesis happens. You can say there are the several causes, like 9 causes, which can be let parthenogenesis happen inside a plant body. Next, we will see here. This is all about parthenogenesis. Okay, so now we will see the second method here. This is apogamy. Uh, Quite similar with parthenogenesis, but there are several different. That's why we give them a new name. So apogamy. Winkler founded it 1908 as a modern one as comparison to the parthenogenesis. It refers to the origin of embryo either from synergist or antipodal cell. I already told you about that. Like they are always involvement of cell, different cell for the making of embryo. So in the case of parthenogenesis, embryo was formed from the egg cell. But in case of apogamy, there will be embryo will be formed from the either from the synergid or from the antipodal cell. That can be the choice of the embryo that what he wanted to form. But both the cell can take part in the formation of embryo in the case of apogamy method. So if we see some main points about that, so it may be haploid or diploid. It can also be haploid and diploid, same like the parthenogenesis. Embryo develop from the cell of normal embryo sac. Normal embryo sac can take place in the formation of the cell. There is no another kind of embryo sac form in apogamy. This using this normal embryo sac word, it will be clear to you when we will study the apospory. So it has been reported in Allium iris and many other plant species. This is very common in the onion. This Allium iris, these are the part of kind of onion species. So basically, this is very common in onions or underrooted plants. In occurs in nature and can also be induced by artificial means it can be happen naturally inside a plant and we can also artificially introduce them. So the types are the same like parthenogenesis. There are two types: haploid one and diploid one. So same if the development of embryo from haploid synergid or antipodal cell, they will be referred as haploid apogamy, and they are also very useful in generating the pure line structures. Second one is the diploid thing. If we are talking about diploid apogamy, so the same in diploid parthenogenesis, but the cell operation different. So the development of embryo from diploid synergis or antipodal cell is called diploid apogamy. So this is also developed from the reduction division, and that's why if there are absence of reduction division, cell have to be diploid and producing the diploid embryo without having the fertilization process. So this is these are whatever the when we are talking about apomixis, if we are talking any kind of haploid one like haploid parthenogenesis, haploid apogamy, haploid apospory, whatever it will be, if they are the haploid one, so they are responsible for generating the pure lines. Okay, or if we are talking about any kind of diploid one, like it can be apogamy, apospory, parthenogenesis. So whatever the diploid phenomena are present here, if they are there, so the main motive of there have to be present for conserving the heterosis. Okay, don't confuse them with haploid and diploid. Their major motto for the haploid one is to generating pure lines, and diploid one for conserving the heterosis, the main the dominant gene which have a phenotypic capacity to perform their actions. So this is all about the apogamy and apospory. Now, apogamy and parthenogenesis. Now we will take a look on apospory. So, in apospory, this is quite a little different from the parthenogenesis and apogamy. What happened here? Actually, the problem is not how the embryo formed from which cell. Their difference is like from what embryo sac developed. Okay, getting my point. Like till now, what we are studying, like the embryo cell forming from the egg cell, embryo sac is forming from antipodal cell, or embryo sac is forming the synergids. But here, this apospory in apospory method, the embryo sac in which these all things are present, synergids, egg cell, polymany, polynuclei, whatever the things that aid cell structure, embryo sac, here from what cell this embryo sac form, and after that embryo sac from the what embryosacal cell the embryo form. So this is a little confusing, but don't get confused because I'm trying to explain you like there the formation of embryo sac is more important rather than the formation of embryo. Okay, so here the formation of embryo sac can be developed from the, or you can say can be happen by two means. We will discuss it. So the embryo then developed directly from the diploid egg cell without fertilization. This is common. This term, apospory, was coined by the D. Varian Power in 1886, and there can be also there is an unusual thing that there can be also the false embryo sac can be formed. Okay, how they will form, we will see it in next slide. So it is two types basically: generative apospory and somatic apospory. This generative apospory and somatic apospory is variant in comparison to haploid or diploid. How? If we talk about the generative apospory, then the embryo sac, okay, the embryo sac here originated from the cells of archisporium. What you have to be take in your mind, the development of embryo sac here will be originated cell of archisporium only. Okay, not from any other cell. This is very definite situation here. The embryo sac should be developed from the archisporium. So this kind of apospory, where the archisporial endosperm development will take place or embryo sac development will take place, that is called the generative apospory. Same reverse in the case of somatic apospory. When embryo develops from the embryo sac that has originated from the cell of either nucleus or integument is known as somatic apospory. So here, from which embryo sac the embryo develop, that embryo sac has to be originated from the cell of either nucleus or integument. Okay. Here, in a generative apospory, the development of embryo sac will be taking place from the archisporial cell. Okay, in generative, embryo sac will be developed from archisporial cell. But in the case of somatic embryostory, the development of the embryo sac happens from the integument or the nucleus cell. Okay, this is the basic difference between the generative apospory and the somatic apospory. I hope you're getting. If still you guys have any problem, just text me after or in a session. We will take these questions. So this is all about the till the somatic or generative apospory. So these are the basically these things are clarify like how apomixis can be different from the sexual reproduction. So now these are some questions you can follow them up in your practices when you are going to take a self evaluation test. So you can do these questions. Okay, these will help you in your further remembering process. I will give you also an LMS and you can go there and do the answer part. Okay, I uploaded it there. Go there and just give the answer here. After these questions, actually, I would like to say that you guys can reference these books like Bhojwani and S.S. Bhatnagar for the embryogeny. Yeah, I think you know that Bhojwani was a very famous scientist which has been done a remarkable work on the embryogenesis. And he, like very few Indian scientists have been working on the botanical studies, but Bhojwani and Bhatnagar was one of them. They did a very remarkable job, mostly on the embryogeny process, means how the embryo developed. The book they published in 2011, Embryogeny of Angiosperm. This is very important. If you go through that, you will find a more of answer. And last is a link for a PDF. This is also good if you are taking a follow-up leads on the Apomixis process. Okay, this is all for the Apomixis classification from the cell involvement. Still, we are leave with the frequency based classification and the occurrence based classification. This is left. Okay, so we will take it into the next class. Until that, if you guys having any problem understanding any topic from this lecture, please ask me later, okay, or ask me in next class. Just to go through these slides again from your LMS and study thoroughly, like in a revision form. Then you can also know what the problem you are facing. Okay.